All right, let's dive deep, like really deep. We're going inside our own cells to check out the incredible, invisible machinery that keeps us all going. And it really all boils down to one huge question.
I mean, think about it. How does something so tiny, it's practically invisible, build its own skeleton, run its own highway system, and then just copy itself perfectly in two? It seems impossible, right? Well, the secret lies with a couple of the cell's most fascinating and honestly kinda overlooked heroes. Okay, so first up, let's zoom out a bit. Picture a cell, but think of it like a bustling city. It's gotta have infrastructure, right? Scaffolding, highways, support beams, just to keep everything from collapsing. Well, that's exactly what the cytoskeleton does for the cell.
Now, don't picture a bony skeleton like ours. This is something way cooler. It's this super dynamic network of fibers that's always changing, building up here, tearing down there. It gives the cell its shape, sure, but it's also the cell's internal highway system, shuttling all sorts of cargo from the nucleus, the city hall, all the way out to the city limits. And this whole network is basically made of three main types of protein cables. You've got your microfilaments, your intermediate filaments, and then microtubules. Now, they all have their jobs, but it's those microtubules that are going to be the real stars of our show today, so keep an eye on them.
So, if the microtubules are the building materials, who's the construction crew? Who's in charge of organizing all this? Well, right at the heart of the cell, we find our main characters for today, the centrioles. These guys are the master organizers, the project managers of the whole operation.
And what makes them so special? Comes down to one single number, 9. Just the number 9. What in the world is that all about?
Okay, here's the reveal. That 9, it's all about the blueprint. Each centriole is made of 9 sets of microtubules, and each of those sets is actually a triplet. So you have 9 triplets all arranged in this perfect circle, like a tiny barrel. We call it a 9+0 arrangement, because there are 9 groups on the outside and, well, nothing in the middle. This design makes them ridiculously strong and stable, the perfect foundation for the cell's most critical jobs.
So let's formally meet these things. What's wild is that unlike a lot of other organelles, the cell's mini organs, centrioles don't have a membrane around them. They're just pure, raw structure. And you almost always find them in pairs, hanging out at a perfect 90-degree angle to each other, kind of like a tiny little L shape. And they're always located right next to the nucleus, the cell's command center.
And that location? Totally not a coincidence. Being next to the command center is crucial because centrioles have the leading role in what is maybe the most dramatic and important process in all of life, cell division.
This is their time to shine. Check out this 4-step mission. First, right before the cell is ready to split, the centriole pair makes a copy of itself. Step 2, these 2 pairs head off to opposite ends of the cell, like setting up posts. Step 3, from their corners, they start building. They spin out this amazing web of microtubules, which we call spindle fibers. And finally, step 4, these fibers latch onto the chromosomes and gently pull them apart, making absolutely sure that each new cell gets a perfect, complete copy of the DNA. It's like a perfectly choreographed ballet.
This whole setup, the centrioles, the spindle fibers, the whole shebang, has a name. The mitotic apparatus. And its entire purpose, its sole reason for existing, is to make sure those chromosomes are distributed perfectly. Seriously, without this process working flawlessly, life just wouldn't happen.
But wait, there's more. Just when you thought their job was done, it turns out centrioles have another amazing gig. They're not just architects for cell division, they're also engineers of motion.
Yep, you heard that right. Centrioles also build the foundations, we call them basal bodies, for things like cilia and flagella. Those tiny little whip-like tails or hairs that help cells swim around. So the centriole is basically the engine block for the cell's outboard motor. I mean, from splitting a cell in two to helping it swim, is there anything these little guys can't do? They are absolutely essential. Okay, so we've laid out the standard playbook for how a cell works. But biology. Just when you think you've got all the rules figured out, nature comes along and decides to break them in the most interesting ways.
Take the nucleus, for example. The rule is 1 nucleus per cell, right? Well, tell that to our red blood cells. They actually toss their nucleus out to make more room for oxygen. Plant sieve tube cells do the same thing.
And then you have the overachievers on the other side of the spectrum. Some cells have 2 nuclei, and some have many nuclei, all coexisting in one big cell. It's just a great reminder of how incredibly diverse and creative life really is.
So there you have it, from the internal scaffolding of the cytoskeleton to the master architects, the centrioles, that make sure our DNA is copied perfectly. They really are the unsung heroes working inside of us every second of every day. And that leaves us with one last and maybe most important question. These tiny structures are the architects of life itself. So what happens when there's a mistake in their blueprints, when things go wrong? Well, that question, that's at the very core of understanding diseases like cancer and so many genetic disorders. It's a humbling reminder that everything we are depends on these microscopic details being absolutely perfect.